Hey guys, we're going to be looking at chapter 1.2 notes, the ultrastructure of cells. For understandings, it's a little bit quicker of a chapter or a section of the chapter. Prokaryotes have a simple cell structure without compartments. Eukaryotes have a compartmentalized cell structure. That means membrane-bound organelles in the nucleus. Prokaryotes divide by a process known as binary fission, and eukaryotic cells undergo mitosis. And finally, electron microscopes have a much higher resolution than light microscopes, which all allows us to see the ultrastructure of cells. All right, resolution of microscopes. Now, according to the book, I did find this hard to believe, but unaided eye can see things with a size of 0.1 millimeters as separate objects. So we use light microscopes to see cells, which allow us to see down to 0.2 micrometers, which is equal to 200 nanometers. It's limited by the wavelength of light. So the smaller the wavelength of light, the better resolution you get. And with light microscopes, the wavelength of visible light, anyway, is 400 to 700 nanometers. And so the common light microscope like we use in lab, its highest magnification is 400x or 400 times. But beams of electrons have a shorter wavelength. The resolution of electron, that E negative, that's my symbol for electron. Electron microscopes is 0.001 micrometer or 1 nanometer. It's 200 times better than a light microscope. So light scopes reveal the structure of cells. Electron microscopes reveal the ultrastructure. All right, prokaryotic cell structure. You should practice drawing these. No nucleus. Most cells have a cell wall outside of the cell membrane, and that protects the bacterial cell, maintains shape, and prevents bursting. There's no membrane-bound organelles. The only organelles they have are ribosomes, and those are free-floating in the cytoplasm. And they are smaller than eukaryotic ribosomes. DNA in prokaryotes is circular and not associated with proteins, and that's referred to as naked DNA. And it's found in a nucleoid. Now, the nucleoid is, it's not a real nucleus. It's just where the DNA is. It's not compartmentalized or anything like that. Cell division in prokaryotes, it's called binary fission. It is asexual. The daughter cells are genetically identical. First, this chromosome replicates and moves to the opposite end of the cells, and you got two daughters that are identical. Eukaryotic cell structure. Nucleus contains the cell's chromosomes, and then single or double membrane compartmentalized sections are called organelles. Each organelle has a particular structure and function to do its role. In biology, structure is always related to function. Its shape determines what it does. Compartments allow enzymes to be concentrated. It allows isolation of harmful substances. It allows pH to be maintained, and it allows for the movement of organelles and the organelles. So the nucleus contains the cell's chromosomes, and those are, in eukaryotes, DNA wrapped around histone proteins. That's to help, because the DNA is so big, it needs to be condensed into chromosomes, so they're wrapped tightly around histone proteins. If you were to unwind the DNA in a human of every cell and unwind it end to end, it would go to the sun and back 200 times. So the rough endoplasmic reticulum, or rough ER, synthesizes proteins that are meant for secretion, so things that will exit the cell. The